Good afternoon, everyone. First of all, I would like to thank the organizing committee for this wonderful meeting. We have no financial interest to disclose. Programmed cell death protein is a signaling pathway for T cell activation and plays an important role in tumor progression by altering the status of immune surveillance. Furthermore, PD-1, PD-L1 inhibitors play an important role in therapy for some refractory tumors. A tumor can be positive or negative for surface PD-L1 expression through several factors, such as genetic mechanisms that lead to constitutive PD-L1 expression, induced PD-L1 expression by the presence of T cells, absence of T cells leading to no reactive PD-L1 expression, and genetic events that preclude PD-L1 expression upon T cell infiltration. PD-L1 expression has been studied in some ocular tumors such as conjunctival melanoma and islet sebaceous carcinoma, but its expression has not been studied in orbital tumors. Mismatch repair deficiency is a form of genetic instability in cancer, characterized by failure to repair DNA replication-associated errors. A defective MMR system leads to the persistence of mismatch mutations across the genome, particularly in regions of repetitive DNA. This study reports on PD-L1 expression and MMR in specific orbital tumors. Our methodology was a historical cohort study of tumors from King Khalid Eye Specialist Hospital, studied in collaboration with King Fahd Medical City in Riyadh, Saudi Arabia, included recent surgical specimens of patients with pleomorphic adenoma, adenoid cystic carcinoma, rhabdomyosarcoma, solitary fibrous tumor, and control orbital tissue. Immunohistochemistry performed on formalin-fixed paraffin-embedded tissue samples and studied for PD-L1 expression using PD-L1 antibody. DNA was extracted from FFPE tissue and used for MMR sequence analysis using next-generation sequencing. As a result, total of 22 samples, 6 samples in rhabdomyosarcoma and pleomorphic adenoma, and 3 samples in adenoid cystic carcinoma and solitary fibrous tumor, and 4 samples as control. In rhabdomyosarcoma, we found that scattered focal cytoplasmic labeling PD-L1 staining on tumor cells was noted in all embryonal types without an accompanying lymphocytic infiltrates, as you can see in figure A, B, and C. The remaining 3 samples were alveolar and were negative. Staining was absent in 5 out of 6 pleomorphic adenoma, and 1 case that showed labeling of lymphocytes in the capsule, as you can see in figure E. Staining was absent in 2 out of 3 solitary fibrous tumor, but 1 case showed focally immunopositivity in figure F. All samples of adenoid cystic carcinoma showed no anti-PD-L1 labeling, and all control samples as well. So this table summarized the MMR sequence analysis. What we found, there are 7 samples that shared the same pathogenic mutations in MLH1. I wish that. And one sample that showed 1 pathogenic mutation in MSH6 gene, and 14 samples that showed negative results in any changes in cancer gene panel. We didn't find any correlation between the mutations genes and certain orbital tumors or PD-L1 expression results. So the studies of PD-1 labeling and MMR mutation in tumor tissues has gained much attention because of new therapies available based on these studies. Based on the presence or absence of T cells and the expression of PD-L1 by cancer cells, a tumor can be categorized into 4 groups: PD-L1 positive, T cell positive, PD-L1 negative and T cell positive, PD-L1 positive and T cell negative, and both are negatives. Tumor responses with anti-PD-1, PD-L1 antibodies are mainly mediated by tumor antigen-specific T cells that had been previously blocked by the PD-1, PD-L1 interaction. In other non-ocular malignancies, where mismatch repair mutations and positive PD-L1 expression was seen, FDA approved anti-PD-1 agents such as pembrolizumab and nivolumab for treatments. These tumors included melanoma, non-small cell lung cancer, metastatic head and neck cancer, and Hodgkin lymphoma. A meta-analysis including 6,800 patients from 51 trials in various cancer types showed that tumors with positive PD-L1 expression had significantly higher clinical response rate compared to tumors with negative PD-L1 expression. MMR mutations and PD-L1 expression has been studied in some ocular tumors. Cao et al. Analyzed the expression of PD-L1 and PD-1 in 27 specimens of primary conjunctival melanomas and found expression on tumor cells in 19% of cases, which was associated with distant metastasis and poorer prognosis. Xu et al. Described PD-L1 expression in 41 patients with eyelid sebaceous carcinoma and positive expression found in 48.8% of cases on tumor cells, with higher expression in metastatic cases when compared to primary cases and higher expression in poorly differentiated group. MMR defect has been studied in 10 cases of sporadic periocular sebaceous carcinoma, which showed no loss of MMR protein expression. In our study, chose to study PD-1 staining and MMR in specific orbital tumors because they tend to be aggressive or recurrent and samples were readily available. All cases studied were primary tumors. PD-1 staining, when present, was limited to focal constitutive labeling in embryonal type, rhabdomyosarcoma, and solitary fibrous tumor, and inducible labeling in less than 5% of cells. Ribas et al. Described nicely the difference between constitutive versus inducible expression, in which in constitutive there is a genetic mechanism through dysregulation of several oncogenes, while in inducible the surface expression of PD-1 induced by the interferon and especially interferon gamma, and that physiological induction lead to evasions of T cells and termed as adaptive immune resistance. So in MMR sequence analysis, we found 2 pathogenic mutations in MLH1 and MSH6 genes. So these results in general suggest that a possibility that these tumors may not be amenable to immunotherapy. Since all tumors with positive PD-1 staining are primary tumors, the staining of recurrent tumors may be different. No clear correlations between MMR mutations and certain type of involved orbital tumors or PD-1 staining. Our limitation, small number of cases, and archived tissue may have affected staining. So in conclusion, based on immunohistochemistry labeling patterns, our study demonstrates lack of PD-L1 staining in many orbital tumors other than rhabdomyosarcoma and solitary fibrous tumor. So this suggests PD-1, PD-L1 inhibitor therapy may not be useful in treatment of these tumors when refractory to conventional therapy. No clear association between genetic finding and pathological presentation of involved tumors. Thank you.
Thank you very much, Dr. Mohammed.